This is a production of Cornell University.
All right, well, thank you, Liz. I apparently have a lot of slides, so I'm going to try to go quickly through them, and I might gloss over a few things, but these are the objectives for the talk. I just want to look at some basically examples that I think illustrate why polyploids are challenging to work with from an informatics perspective and a biological perspective. I want to take the time to use those examples to just look at some of the tools and resources that we've developed or adopted from other groups and adapted to our needs, using them for our particular research community, but hoping that they'll find more broad adoption. And the last is probably the most audacious of the 3 bullet points, but I've been working on abstract algebra as kind of a side hobby, and I want to try to make a case that it's actually of relevance to understanding how complexity develops out of sort of nonlinear multiplicative relationships like those that come out of putting 2 genomes together. So we'll see how that goes. All right, so just a quick kind of overview of polyploidy in general. It happens all the time, seemingly, in plants. It's known to happen in some other systems, but way in the evolutionary past. So if you work in plants, you know all about it. If you don't work in plants, it's kind of a weird little niche. There are a lot of distinctions to be made about types of polyploidy and how many genomes have come together at once and how recently it was. And those are all important for thinking about the actual approach you're going to take with a particular analytic solution to a problem. I've heard that the thinking is that one of the reasons why plants are so tolerant of polyploidy is that most of them don't actually have sex chromosomes, and that facilitates the combination of genomes. They exhibit there are challenges to the plant associated with actually working out what to do with having multiple genomes. But after they've done that, they tend to be a little bit more resilient. And so a lot of the crops that we work with certainly have come from polyploid events, possibly because the gene redundancy kind of gives you new opportunities. And we'll look at some of those questions a little bit further on. And they tend to occur in extreme environments, partially because those environments tend to induce polyploid formation, but also the thinking is that they are, because of the built-in redundancy, they're better able to adapt to such conditions. As I said, there are a lot of different types of polyploidy. So one distinguishes between allo and auto depending on whether or not the 2 genomes that are being duplicated are the same or different. There's a type of polyploidy that has to do with somatic cells. Doesn't actually lead to new species, but basically mitotic events, not actually separating the results of genome doubling into separate cells. So that's Endo polyploidy. And then distinctions are made, in terms of how recent the polyploidy event is and how far along the evolutionary trajectory it's gotten so that it's effectively become a diploid genetically again. And depending on whether you're an auto polyploid or an allo polyploid, that introduces, auto polyploids basically all of their chromosome, well, the homeologous chromosomes, as we call them, can potentially recombine with one another. And so that introduces new possibilities in terms of the genetics that need to be thought about carefully. So as Liz mentioned, that we're interested in comparative genomics. We've developed a tool for basically looking at gene content across species. Each of these glyphs represents a gene. It's colored in terms of gene families they've been associated with. And when you're dealing with say, simple species like primates and mice, you have nice, fairly simple relationships between the gene content of the various genomes. One-to-one relationships in general with occasional presence-absence or copy number variation. It's pretty easy to just pick out. But when you're thinking about comparisons between genomes, each of which has a kind of separate duplication history, things become a lot more complicated. And so here we have a few different species that have different polyploidy histories, some ancient duplications that they share, and then more recent duplications that have been separate in the different species. And so we've got a little track of a soybean chromosome at the top. We can see that it's been through two whole genome doublings. One of the tracks is much more similar. That's the more recent duplication event. And then you've got some more ancient events that are closer to one another because their doubling resulted from that more recent event, but further away from them. Even something that you tend to think of as a small genome like Arabidopsis has, again, chromosome doublings or whole genome doublings involved, with a lot of gene loss afterwards. So you can easily see here patterns of fractionation of the subgenomes that resulted from these whole genome duplication events. Tomato actually went through a hexaploidy event. And this tool, basically allows you to dynamically query databases for tracks that have a certain gene content associated with them, and then pull in from disparate databases tracks that seem to have similar gene content in terms of the gene family assignments, and then ultimately ask questions about the evolutionary history behind genes that have been aligned through synteny together and their phylogenetic relationships. And so you can see that out of those four, for that particular gene, which was from a abscisic acid responsive transcription factor family, out of the soybean homeologs, 3 of them retained that gene, and it was lost in one. Similarly with tomato, 2 were retained, 1 was lost, and Arabidopsis also retained 2. So this gives us a nice way of exploring relationships among the genes, but the question, that still is difficult is, well, what happens to function as these things have duplicated and then gone their separate ways? And that's not so easy to resolve. So we're also interested in, well, some polyploid species like soybean are economically valuable and have a lot of research that goes into them, have their own database resources with curators pulling in information from the literature. Can we make use of comparative genomic techniques to essentially take something like cowpea, which does not have that more recent whole genome duplication event, and figure out, okay, do I have any, say, SNPs associated with seed weight in regions in this genome, which I know has sort of more or less 2 copies of genes for every copy of a gene here, and find out, well, yes, there is a region where I found an association between the trait of interest that is similar to one of the homeologous regions in soybean where they've also observed similar variation. So that's obviously a nice way of taking candidate genes and figuring out, well, have I seen evidence in some other species that changes to this gene actually affect the trait of interest? All right, so here comes the tough part, making the case for polynomials. So just as polyploids involve multiplication of kind of independently behaving linear genomes, polynomials are kind of like that in that you're taking together linear factors. And if you think about it in this direction, it's pretty easy to go through and use the distributive property or things that you learned. I was in Lansing High School, so I learned this from Mr. Cotmell. You do the first outside, inside, last trick and figure out exactly how to do your factorization. And it's essentially no different no matter how many factors you have. You're doing combinatorial distributive multiplication, and you're essentially taking the roots of the equation and seeing them come out in the coefficients in kind of interesting ways that these are what's referred to as the elementary symmetric polynomials, which means that it doesn't really matter how you permute them, you get the same result. And this ends up being very important in terms of theory involved in what it actually means to solve a polynomial equation. So, there are numerical techniques that allow you to approximate solutions, But what algebraic people want to do is to say, what is the actual algorithm? Can I find a formula like this old guy you probably remember, the quadratic equation? You know, it's not exactly simple, but it's fairly straightforward. You probably remember how to get there. You either memorized it or you learned completing the square and can rederive it. I mean, it's not too bad, but once you go even to a cubic equation, things get really a lot harder. And this is Renaissance mathematician Tartaglia, who figured out how to deal with these things by first performing a little trick called making a depressed cubic, and then eventually getting to a formula that in some sense is analogous to the quadratic equation, but it's a lot more complicated. And you get also interesting things with higher degree equations, like what they call casus irreductibilis, which basically means that you can have three real valued roots, but if they're not rational, you can't actually represent them with this kind of representation without involving imaginary numbers, even though the results that you get are not themselves imaginary. So as you start to increase the number of factors of things, life gets interesting. And going even further, it was eventually demonstrated after people had solved the cubic and the quartic. They were working real hard to try to solve the quintic, and eventually, through the work of Abel and Galois, they discovered that in fact you can have fifth degree equations and higher where you simply can't represent the roots in any way using that sort of representation. And whether or not you can get to some sort of a representation like that is basically dependent on this notion of symmetry among the roots and how the coefficients impose constraints upon the roots, whether you can represent relationships among the roots starting from what they call the field of the coefficients. So an example of that would be cyclotomic polynomials, which sort of distribute themselves in the complex plane around a circle. They have a kind of a very restricted form, and it can be shown that any cyclotomic, you can represent the roots in terms of these kind of specialized quadratic, or not quadratic, but radical towers from the field of the coefficients. Okay, well, there are other times when taking together a bunch of disparate things that are simple to deal with, relatively simple individually, and you try to put them together and things get complicated quickly, even when you're doing kind of the multiplication aspect of things. So this is a project from which many of the following examples are taken, an NSF-funded project that we've been working on to try to take a lot of disparate information resources and unify them in some way. There are some technical challenges in this. There are many sociological challenges to actually getting something like this to work. But I won't go into that too much, other than to say that many of what I show would not be possible without our collaboration with many of these groups. So here's my first example. Cultivated Peanut. It's a recently formed allotetraploid that was formed from a few diploid wild species. So the original genome sequencing was done on the diploids. They kind of recognized early on that the diploids were close enough together that it would be challenging using the sequencing technologies available at the time if you were to just sequence the tetraploid directly to figure out which parts went with which subgenomes. So they approached it by figuring out which were the actual progenitor species, and actually one of them is there's only one accession of this species out there, and they believe that it was transported down into the range of the other species by prehistoric humans, allowing this hybridization event that eventually led to the cultivated polyploid. So you can see it sort of there amidst the range of the other species. So you can kind of synthetically create what the amphidiploids, which are, I think it's just a fancy way of saying an allotetraploid from these things that were created synthetically. And so that's the strategic approach. And you can see that there are some, if you're comparing chromosomes, corresponding chromosomes from the two, They are different structurally in some ways, but there's kind of a clear one-to-one correspondence between the two chromosomes. So one of the interesting things about Peanut is that because of this extreme genetic bottleneck that it went through, they think that there was essentially a single hybridization event that ended up propagating into all of the cultivars that we have today. So it's very non-diverse from a traditional allelic sense, but phenotypically there's quite a bit of diversity associated with things. And so this was kind of a question for people as to how is it possible for such a limited genetic base to generate all of this phenotypic diversity. So when they, this was actually something that they began to see when the diploids were sequenced and compared to data that was available for the tetraploid. But essentially what's happening is that the two subgenomes within the cultivated tetraploid are interacting in different ways, such that in some cases you have a segment of one subgenome that's replacing the other ones. This is just a representation kind of showing copy number of the two different homologous genomes. So here we have at the end of chromosome 14, I think it is, from a particular variety of peanut, a place where the B ancestral genome exists in two copies and the A has been replaced with it. And other regions where A has been lost and B has been retained, or the other way around. So essentially what seems to be going on to a large extent with Peanut is that different patterns of this interactivity between the subgenomes are generating this type of phenotypic diversity. And they were actually able to show that they made one of these synthetic crosses and kept it going for a few generations, and were actually able to reproduce some of this phenotypic diversity within that pedigree. So I thought, well, okay, if that's the case, we've got the TIFF runner genome sequenced. Could we actually see what they were showing in that diagram using some of the tools that we've produced? So here's that so-called genome context viewer again, showing us the two homologous segments of cultivated peanut and then the progenitor genomes. And at this level, you can see some cases in which it seems as though the gene content of the two homologous segments is reflecting the B genome rather than the A genome. But it might be a little difficult to argue that couldn't also just be vagaries of annotation effects or misassemblies and things like that. But if you go ahead and select, say, the gene right at the end of the chromosomes, and then look at it within the context of the tree, you can actually see the pattern fairly clearly in terms of the sequence level relationships that indeed, in this case, the B genome genic sequence is much closer to the two copies from the tetraploid than the other genome sequence is. And you can interact with this tool in ways that collapse down the tree to just those genes of interest, and then see that reflected in the multiple sequence alignment. You can see various places at which the A genome representative is indeed different from the others at a number of positions. But of course, if we take another gene from that same region of the genome, it might reside in a gene family that's a little bit more complicated. So here again, we've got the B genome with the two tetraploids taken from the same region and behaving similarly in terms of tree topology. But there are other subclades of this tree with different sorts of patterns associated with them. So you can pretty easily, using this tool, again, collapse down to the set of genes that may be of interest in your particular context, and then go in the reverse direction to say, well, let's look at their genomic contexts and figure out, not starting with the premise that you're looking at things that are all synthetic, but asking the question, how would I group these things given their kind of gene family neighborhoods in such a way that I see the synthetic relationships that exist among members of this gene family. So this, I guess this is the next gene down from the end of the chromosome that I chose. So this would be our chromosome 14 genes. And then this is the other gene family, or other clades that you can see has kind of a more complicated picture in terms of the gene duplications within that region. And again, you know, some of the aspects of the way that those sequences appear in the tree may be driven by our annotation sorts of artifacts, but we believe that these sorts of tools are valuable for people, you know, who want to ask particular questions that will enable them to browse the data fairly effectively, even in the context of having these kind of fairly complicated relationships between and within plant genomes. So we're really excited about our development of this tool, and hoping that other groups will eventually start to pick it up too. All right, so moving on to a different example of challenges associated with polyploidy. This is a genome sequence project that we've been involved with for a number of years, actually more years than I care to admit, but it's been a bit difficult going, both for technical and funding reasons, I guess. But you know, so alfalfa, cultivated alfalfa, is a species that is an autotetraploid, and making life more complicated still, it's an outcrosser. So there's a lot of diversity within the genome itself. It's related to Medicago truncatula, which is a kind of a modeled species for the legumes. And so, you know, if you look at something like Kamer frequency spectra in Medicago, you get a really clean result showing you a sort of a Poisson distribution of your Kamer frequencies, which is what you would expect from single copy sequence content classically. And you can model things about genome size and heterozygosity, which effectively this has none, using this type of setup. When you try to do the same thing with sativa, things get a lot more complicated in terms of the set of Kamer peaks that you're dealing with, and your attempt to fit models of those kind of mixed model distributions of Kamers present at different copy numbers that you're sampling from a large pool that you've generated from whole genome shotgun sequencing. Well, so one thing that we did in that context, kind of similar maybe to the peanut genome, there was a diploidized version of cultivated alfalfa that had been taken from a tetraploid and made into a diploid. And we thought, great, this will be simpler. We can start with that and then leverage it to help us with when we're dealing with the tetraploid itself. All right, so we had a lot of different data sets that we had available, or you know, we started thinking it would be easy with Illumina sequencing, and then as that proved not to be the case, we moved on to different types of data. So PacBio sequencing, I'm not exactly sure how familiar you guys all are with next-gen sequencing, but you know, there are a number of different technologies, not all of them sequence-based, but they all have kind of their strengths and weaknesses with respect to getting at complex genomes. And so we've tried a number of things. And one thing that's kind of a little bit unique about autotetraploids in terms of using linkage maps is that you do your linkage mapping typically using, say, single dose heterozygotes. So they sort of behave from that perspective like diploids, where it's this or that segregating, and you can just keep your eye on the informative haplotype and figure out how it's segregating with other informative haplotypes from the same linkage group. So I maybe trying to stretch that algebraic analogy, but you know, as you think about roots of numbers that are more than just the square root, where maybe you think about positive or negative, as you go to higher roots, you have to think about all of the different directions in the complex plane that you could be talking about when you're talking about the root of a number. So things with genotyping get more complicated as well. Just trying to get a handle on what the material was in terms of, you know, what is it that we're trying to assemble here? Do we essentially want to get 8 chromosomes like Medicago has, even though we know there are 32, but for each of the 32, 4 of them should be in some sense, well, homologous to one another, but at some level of diversity from one another? Or do we really want a 32 chromosome sort of pan-genome within the context of our single cultivar of alfalfa? You know, and in some sense, every time you create a cross of alfalfa, you're going to get a different combination of these things. So in some sense, it's kind of quixotic to try to get the haplotypes, but if you're clonally propagating it, you'll preserve them at least, and you can ask questions about, you know, is this technology suitable for understanding issues of haplotype phasing across long distances? So probably not surprisingly, you know, we found that short read assemblies were compressing things down a lot, more towards the expected haploid genome size, but there was a lot of fragmentation. You know, if you think about your N50s relative to chromosome sizes, they're not looking very good. Even with the long read technologies, um, the contig sizes that we're getting out of those are very small, not as small as the short reads, but at the level of smallness that it's even challenging to kind of take some of the other technologies and apply them to them. And the other thing is that the long read assemblies, you know, were definitely producing genomes that were closer to capturing several of the haplotypes, not maybe all 4, but you know, and even in CADL, which we had approached as a diploid, thinking it would be simpler. Well, it was in some sense, but still, there's a lot of heterozygosity there, and enough that with the high error rates combined with the heterozygosity, there are a lot of issues with actually dealing with long reads and figuring out, you know, well, is this just a repeat? Is this just a highly errored sequence, or is it actually a divergent haplotype? So anyway, when we finally kind of took one of the long read assemblies as our base, then we started exploring kind of higher order genome scaffolding technologies like optical maps or high-C of various flavors. And one of the companies we worked with, Phased Genomics, their algorithm basically, you would tell it how many chromosomes you wanted to get out, which in some ways seems a little bit of a danger sign, but you know, we could tell them, well, we would like to try it with 8 chromosomes, we'd like to try it with 32, and you can see that there were some significant differences, and I will say not differences that always made a lot of sense to us in trying to do that. I talked a little bit about the Kamer frequency spectra. So, you know, depending on how what size you choose your Kamer at, you will either pick up stuff that is effectively single copy across all the homeologs and present at higher frequencies, and then smaller peaks representing things that are single dosage or double dosage. I think that you know, this progression as you get closer to using longer Kamers and showing that your peaks, your higher order peaks, effectively are being erased, you know, is effectively showing us that they're within about 71 base pairs, you know, you're going to get 2 SNPs that actually differentiate between all 4 haplotypes. So it's quite a complex genome to work with from an assembly perspective. But when you look, so these are the same, so using 25 MERS as our basis, we're using kind of the Kamer frequency plots against the actual genomes that we've assembled and looking to see, well, for Kamers that are in this peak, do we capture them once as expected or twice? And well, I guess this is actually this is the peak of 4x, so it's shared between all 4 haplotypes. So you might expect that to be captured at 4x. And you know, again, it's not surprising that what you see is that the short read data tends to pick up this, you know, single copy stuff, and you know, some of the haplotypes are not being represented, so they're at 0x. But it's not really a simple picture, and even within CADL, where the Kamer frequency plot looks a little ragged because we don't have the same depth of coverage because we're using PacBio data, which isn't as deep, and you can't really use it prior to correction, which is one reason it becomes less deep. Things look complicated again, as you would expect with a heterozygous genome. So BUSCO analysis is something that a lot of people do to kind of assess how well their genomes have done in terms of capturing genes that are expected to be present in single copy. And of course, whether or not you expect it to be single copy is again kind of relative to your expectation for the ploidy level and the amount of time things have had to become single copy again following a whole genome duplication event. And in terms of genic content, again, the short read technologies in general, well, these are different now assembly algorithms being applied to short read or long read. And again, you can see that short read technologies are doing an okay job of picking up gene content, but just basically making a single copy, whereas the longer read technologies are picking up 2 or 3 copies of these things. And when you look at genes from Metacargo truncatula, just across the board for things that you wouldn't necessarily expect to be in single copy, you get a somewhat similar picture, but definitely more things appearing in multiple, 4 or 5 or higher levels of ploidy. So not a real change, but kind of tendency to be able to pick up multi-copy gene families, I suppose. All right, so we had we have the Metacargo truncatula genome, and prior evidence made from genetic maps within sativa showed us that essentially the 2 genomes are collinear, despite the fact that even at the haploid genome size, one is quite expanded, about almost twice as big as the other one. There are a couple of interesting things that turn out not to really be about differences between sativa and truncatula, but the particular accession that was chosen for the genome sequencing of truncatula had some peculiarities, and they show up when you compare it to other truncatulas. But when you essentially try to use truncatula as your basis, you would expect that it would do a pretty good job of showing you collinearity. And depending on how fragmented your assembly is, genome dot plots are a nice way of showing relationships between different genomes. But when you see something like this, where the labels of one axis are so dense that you can't even read them, it basically just means that you've got a lot of degrees of freedom that you have to make things look like diagonals. And if there are real structural events going on between your genomes, you're not likely to see them. But as you start applying more aggressive scaffolding technologies, then you get the chance to be wrong. And this is an example of some of the things that were coming out of the high-seed data, where they're doing a pretty good job of kind of figuring out which chromosome a particular scaffold belongs to and getting it in the right neighborhood. But effectively what they're doing, even when you tell them to kind of produce 32 chromosomes, is not really give you 32 chromosome haplotypes, but give you each of the 8 chromosomes in 4 pieces, where each of the pieces kind of represents a strange little jumble of all the haplotypes that you had for that, because the high-seed data isn't really able of resolving these kind of haplotype phasing issues, at least not in the hands of the algorithm that was applied to it. So just zooming in on this a little bit more on one of the chromosomes, you can start to see, on the one hand, probably some assembly artifacts that are coming in from some of the scaffolding technologies. I would not expect this to be a real relationship between a alfalfa scaffold and truncatula. You can see another one of those sort of crow's feet representations there. But you can also clearly see, we are picking up fairly good representation of 2 haplotypes along, in many cases along wide stretches, and in some cases it's a little bit more fragmented. Again, if we take our original non-scaffolded, just long read technologies, you can start to kind of trace a picture, but you have a lot of degrees of freedom to make this into little, sort of line segments that are showing you collinearity. As you apply something like optical mapping, these lines become much longer and more coherent. And these seem to represent haplotypes, but there's still kind of a question about can you really trust that the phasing that this is sort of saying that you have along your haplotype is actually accurate? Because a Bionano map doesn't actually have sequence data associated with it. It's just a kind of a pattern, which we'll see in another slide. So this is the kind of a gamish of haplotypes that the Hi-C data is producing for this region. Again, probably in the right general vicinity, but it's certainly not making a single coherent haplotype as instructed by the 32 chromosomes. And then if we apply a Hi-C plus Bionano, which kind of allows you to make breaks where necessary, and one limitation of the non-sequence basis of something like an optical map is that your scaffolds kind of have to be a certain size to begin with in order for them to get a purchase into the genetic map, or sorry, no, the optical map, which I'll show in another slide. This then again is using the same tool to look at the haplotypes that we've derived from that region. And here is just kind of the crow's foot sort of thing that we were seeing in the dot plot looked at from the genic perspective and turned on its side for CADL. So that's what I would consider to be an artifact scaffold. But we also see some kind of interesting things within our alfalfa blocks where, this is just kind of a contig that the optical map was not able to place, probably because it wasn't long enough and didn't have enough restriction sites to really create a pattern to put it into place. But you can basically imagine that it fits right in there. Here's another example where you have a little region. We didn't happen to pick it up using the kind of Synteny-based way of looking at things that we're using here. But another kind of interesting question then, again, along the same lines about, well, what do people who want to use an alfalfa genome really want out of a reference? The tendency would be to say, well, let's just choose the longest haplotype and kind of do a minimum tiling thing and choose the most contiguous and least redundant path through those dot plots that we can. But there does seem to be a lot of evidence that there's gene loss between the different homeologs, as some polyploid theory might lead you to believe could be the case. And so, if you just chose one representative homeolog, then you might be missing important gene content that was actually present on another one of these copies. And so, as I sort of alluded to the fact that you've got kind of a pan-genome for the price of a single genome with alfalfa. And I'm thinking, eventually we might come to the place where instead of thinking about single reference genomes, we think about consolidated references that could basically take all the 4 homeologs, kind of abstract them out into a single representation that was maximally useful for somebody actually trying to do things in alfalfa. All right, so optical maps turned out to be a bit of a surprise for me. I was thinking that they wouldn't really have enough information to be able to distinguish the homeologs, but what we were seeing is that when we took the original contigs and then started placing them up against the scaffolds that it would produce, we saw that they were kind of breaking the contigs and distributing them across different scaffolds in a kind of a pattern that suggested that maybe what was going on was it was trying to make a haplotype consistent phasing. And so this is kind of what optical maps look like. You sort of do an in silico digestion of your sequence data that you've produced, wherever the enzymes that you're using, you find their cut sites, and you look at the pattern that this would create, and then you look at all of the labeled molecules that you've put together using assembly-like techniques, and look at the patterns that they show. And so in this case, we can kind of see some clear regions where maps created using different enzymes are showing places where you get a very clear crossover between the sequence scaffold showing a pattern that's associated with this map up to here and this map up to here, and then switches over to not matching this one, but matching this other one, which hadn't matched before. So that seems to be pretty good evidence in my mind. These things are actually heterozygous enough that the haplotypes are actually fairly well distinguished, even when you don't have the sequence information to deal with. So finally, we're trying to make use of these genetic maps, and even that's been a bit of a challenge because the markers that were developed using one of the short read assemblies, and so those short read assemblies kind of represent little mosaic haplotypes, even within the context of fairly short stretches of flanking sequence. So what you'd kind of like to do is to say, well, I know this was a SNP chosen because it had 3 copies and 3 of the homeologs and 1 copy of an allele in the other one. So what I really want to do is establish, what's the informative allele, which linkage group did it go to, and do within the context of the linkage-phased sort of representation of those SNPs. Am I also phasing those particular alleles within the context of sequence analysis? And so this is showing us a dot plot where the different 32 linkage groups genetically are being represented, and you can see that it's fairly collinear with a little bit of noise off the diagonal, showing where a particular allele really ought to be in this linkage group that was actually put into a scaffold with something else that it doesn't really belong to. And then finally, getting down to the actual sequence level, the haplotypes, it was a bit of a surprise to me to see that at the end of the day, even though we didn't really end up using CADL to be kind of a substrate for working on the higher level putting together of the tetraploid genome, when you start looking at the haplotype structure that you have in terms of the different things appearing in these 2 genomes, they're actually, it seems as though you're seeing, in some cases at least, similar haplotypes resolving out of those 2 genomes, which to my I don't know that much about the history of CADL, but I don't think was actually really derived in any way from this material. So that was kind of a nice result. And then up here, we're kind of showing what a multiple sequence alignment tool for whole genomes kind of does when you try to give it things that actually have multiple genome copies, but you're just sort of saying, well, take this species and align it here, and I expect there to really be one region, but it's finding that you've given it several copies of this region with different characteristics. It's kind of just mashing them into one consensus-like thing that is much more nicely resolved down here. All right, so this is the kind of the last example, and this represents more recent work that is actually just a proposal that was recently submitted that we've been generating a lot of preliminary data on. And so, people have been looking at functional genomics, omics data in bulk tissues for a long time now. And it's great, it's powerful, it provides us with a lot of information, but one problem is that, in fact, you've got a genome that in some sense is the same in all of these parts of the plant, but it's doing different things. And, when you aggregate things together, you can obviously miss interesting things that might be going in the parts, and sometimes you can just get patterns which are actually the opposite when you look across the aggregate than the things within the meaningful subgroupings that you had might have related, and that's called Simpson's paradox. So the question is, well, if we start looking at things at finer levels of resolution within a system, a multi-cellular complex system, will we be able to answer questions that we couldn't address when we were just looking at things in the aggregate? And this is work that Marc Lebeau has been spearheading. So just kind of giving a little bit more substance to what I was saying, the root systems of these various species, although they have, in many cases, the same cell types, where cell type, I'll just let you decide for yourself what that might actually mean. They look pretty different in terms of their structure and the number of components of different types that they have. So you can kind of easily imagine that when you're averaging out in kind of different weighted averages across these different pools, you would not really be able to compare all that easily across them in meaningful ways if you wanted to say, look at, even if you had done a kind of a carefully controlled experiment where you defined, what is the section of the root that is homologous in Arabidopsis to Medicago, some of these other things, you may just be limited in terms of the actual biology, in terms of what averaging is happening. And so when you try to do comparisons in terms of evolutionary questions, you just might not really have great data to work with. So recently, as I'm sure many of you are aware, technologies that have been around in the animal space for a number of years have just begun to be applied to plants. It's not that we're slow on the uptake, it's that we've had real challenges in terms of cell walls and the ability to actually take plant tissue and dissociate the cells into, say, contexts in which you can actually do single cell biology. But within the last year, a number of papers have come out, including John Schiefelbein, who was on the previous slide, who's looked at Arabidopsis root development for many years. He published a paper using protoplasting on Arabidopsis root cells, and basically created a Arabidopsis root atlas from that data, as a number of other groups have been looking at. And I was recently made aware of an effort that's, there's a workshop happening soon to kind of start thinking about what it really means to generate a plant cell atlas, similar to the sorts of atlases that are being developed in, say, human and mouse and other systems that are a little bit more mature in terms of these technologies. So I think it's a really exciting area, and something that I'm hoping that we'll get funded to work on. So Mark's approach has been a little different in that he's working with single nucleus data rather than single cell or protoplastid single cell data. And a number of the possible advantages to doing that are enumerated here. One of the ways that I relate this to the basic question of polyploidy, and I thank Jeff Doyle for turning me on to this question about what happens, transcriptionally with cells that have different endopolyploidy levels. A lot of these platforms will have different biases in terms of, cell sizes that they're able to actually incorporate into their microfluidics or their droplets. And so that's at least somewhat minimized with the use of nuclear data. But I think at the end of the day, it's probably just the fact that you don't have to tune your system as much when you're trying to go across different species working with the nuclear isolation protocols. So hopefully we will be at a point soon where we can actually engage in some of the comparative functional omics that we are hoping that these technologies will really start to bring to bear on these questions. And we're thinking that a lot of the biological questions, may actually have slightly different kind of takes that you get when you're restricting to yourself to what's going on in the nucleus. But that could be viewed, in some cases as an advantage rather than, possibly just a difference. And there are some real challenges too. When you're going to the single cell level, you're already faced with challenges with really low amounts of starting material, and when you're limiting yourself to only what's going on from the nucleus, it's even more challenging. But one basic question with using the single nucleus approach is, well, can we even get a picture of these systems that's comparable in some ways to what's starting to come out of the single cell studies? And so these are the kinds of pictures that are created by some of the informatics systems associated with single cell. Essentially, each data point is an individual cell, and they're being spread out into this kind of two-dimensional projection. You can think about this sort of like a PCA, except PCAs are kind of designed to look at the dimensions that maximize variance across, well, what are the dimensions across which most variance occurs. These techniques are a little bit more in tune with looking at complex sorts of relationships where neighborhoods are more important. T-SNE was the one that people kind of started out using. I first heard about it a couple of years ago at PAG, and already it's been kind of started to be deprecated in favor of more sophisticated techniques. But in any case, you can see that if you do kind of a co-clustering that takes into account kind of possible batch effects between the different experiments, and then you project them into the same space and then separate out the single cell dots from the single nucleus dots, you get a fairly coherent picture in terms of the overall structure where different areas are populated by both cells and nuclei. But there are some things that we're picking up that don't seem to be present at all in the single cell data. So that could be a case in which there are just certain cell types that are biased for whatever reason against being picked up in the protoplasting approaches that we're able to pick up with the nuclei. So it's it's fun to look at these plots sometimes. 2 minutes, okay. You can see sort of patterns, but at the end of the day, what you really want to do is to sort of figure out, well, what are these cell types? And so if you're working in Arabidopsis, you have marker genes that have been established for cell types over many years, and you can use the patterns of expression to kind of tease apart what these clusters likely represent biologically. Similarly, in soybean, which is Paleo Polyploid, we're able to sort of do a similar thing, figure out what the clusters represent, and then start to look at effects of treatments like inoculation with symbionts and show differential expression between the genes that are in the same cluster but being affected by the treatments differently. And so I probably won't have too much time to talk about this, but Jeff and I have worked since the early days of NGS sequencing on questions around, well, when you get new polyploids being formed from hybridization of diploids in these polyploid complexes, essentially. You call them all toe and tella, but they're really different species, different genomes, different combinations of genomes. What actually happens with the expression of these homeologs when you put these genomes together? And can you access those questions using some of these sequencing technologies, where sometimes you see, one homeolog effectively gets suppressed and the other one is sort of dominant. Sometimes you get things like subfunctionalization. And so we're trying to take the single cell data and basically ask those questions by taking the data from the single cells that's been aggregated across the clusters and then defining indices that describe, in terms of gene pairs related to one another through homeology from ancient whole genome duplication events, what are the sorts of patterns that you can see? So this is a case where for this particular gene pair, sometimes one gene is the dominant partner, sometimes the other gene is the dominant partner, and sometimes you don't see any bias or you don't have enough read count data to really see significant bias. Sometimes you see clear preference of one homeolog over another. And then similarly, are different tissues different in terms of the ways in which they use different homeologs to become the cells that they want to be through the developmental processes. So finally, in addition to transcriptomic data, we're using ATAC-seq data, which really is looking at chromosome structure and how the chromosomes remodel themselves through interaction with the histones to try to make gene expression possible. So there are some integrative techniques that have been developed similar to what you might use when you're trying to integrate, say, a data set from one species to a data set from another species. You're kind of making some assumptions about what expression would look like with respect to the accessibility of genes that are being expressed, integrating that way, and then trying to figure out what the clustering is, And then you can start asking questions about differential accessibility over the attack-seq data aggregated across your clusters, attack-seq being even more sparse than the RNA-seq data is. So here are the people who I haven't already mentioned in the course of this talk that I'd just like to acknowledge on the various projects. And I'll take questions.
The next class. Are there any questions? Time for one or two questions.
Fascinating talk. Thank you very much. I guess I had a question about kind of the prevalence of those presence-absence variants you're seeing in the Tetraploid alfalfa. So one of the things that ended up happening is autotetraploids end up developing a large amount of genetic load, and I'm kind of curious about what the prevalence of those presence-absence variants are, and whether or not it's missing pieces that are actually contributing a lot to that.
Right. Yeah. So
the question is
right. So I would say it's not a question we've looked at carefully yet. We haven't actually done a gene annotation of the tetraploid, mostly just for reasons of running out of funding. So the data I was presenting was actually taking the truncatula genes and just projecting them onto the tetraploid and showing it from that perspective. So there's a little bit of guesswork involved with that. But in terms of the seeing fairly large deletions present in genic regions, when you look at individual examples, I would guess that it's not uncommon to see those types of mutations, changes being maintained within the gene pools.
any other questions? One question?
Sure. So what are you finding in terms of the similarity in cell clusters between soybean and Arabidopsis, given the morphological differences you showed in the groups?
Right. Well, that is a question I unfortunately can't address at this point. Mark has been very active in sort of taking the marker genes that he's studied and just establishing the identity of the cell clusters. And I think our soybean data isn't yet that. It's not as good as our Arabidopsis data. I said that the nuclear data would involve less protocol tweaking, but I think there is some protocol tweaking just in terms of basic sizes of nuclei for the larger genomes. That he mentioned something about centrifugation being an issue. So I'm imagining that there are some bursting issues that he's still working out.
Andrew,
This has been a production of Cornell University, on the web at cornell.edu.